Hello, everyone, and welcome to today's Journal of Drugs and Dermatology webinar. JDD would like to thank Crown Laboratories for supporting today's presentation. And now I'd like to introduce Dr. Mona Alcom, Director of Clinical Development and Medical Affairs for Crown Laboratories. Welcome.
Good evening, everyone, and thank you for joining tonight's webinar. Novel Biome-Care Technology of C. acnes subspecies defendens demonstrates health and aesthetic benefits for facial skin. My name is Dr. Mona Alcom, and I'm the Director of Clinical Affairs and Medical Development at Crown Laboratories. I will be moderating this evening's webinar. As Crown Laboratories continues to emerge as the leader in microbiome skin health this year, we are excited to share these types of educational opportunities with the medical community to elevate your thoughts beyond traditional skincare to the benefits of microbiome care. Tonight's presentation is part of Crown Laboratories' ongoing continuing education series. Tonight's speakers include Dr. Thomas Hitchcock, Chief Science Officer for Crown Laboratories. Dr. Hitchcock oversees clinical development, medical affairs, and research and development for Crown and its subsidiaries. He was also the founder of Therapeutics, a skin microbiome biotechnology company, and served as its chief executive officer until acquisition by Crown in 2019. In coordination with his work at Crown, Dr. Hitchcock is an adjunct professor of the ETSU Department of Biomedical Science. Dr. Hitchcock has more than 15 years of research experience, including leading the research we will be discussing today. In addition, we are thrilled to have Dr. Deirdre Hooper joining us this evening. Dr. Hooper is a board-certified dermatologist who specializes in cosmetic, medical, and laser dermatology. In addition to her private practice, Dr. Hooper is an associate clinical professor in the Department of Dermatology at both Louisiana State University and Tulane University. She serves in a leadership capacity for various distinguished dermatology, cosmetic, and laser societies, including serving on the board of directors for the American Society of Dermatologic Surgery and Women's Dermatologic Society, as past president of the Louisiana Dermatology Society, and as chair and member of numerous committees, one of which is the Scientific Advisory Board for Crown Laboratories' skin microbiome initiatives, some of which we will discuss today. So let's get started. Dr. Hitchcock?
Thank you. It's my pleasure to be here, everyone, and we are very excited to present our clinical evidence for this technology today. This is the first time we publicly shared this. And so I wanted to get started by giving a brief background on the scientific basis for which the clinical research we're going to present to you today would determine what strain we were going to use, because as the title may surprise a lot of people that might be watching this as to why we chose this particular subspecies of bacteria for a Biome-Care platform. But for those of you who watched our last webinar, these first slides may seem repetitive, but don't go anywhere, because at the end, we're going to show clinical data, not at the end, but midway through, we should start showing clinical data that will present that is pretty compelling. So let's start by there you go. So I'm going to start very briefly. While most of you watching are likely already aware of what the microbiome is, though there are some people that are still kind of not really sure how to define the microbiome and why it's important. So very briefly, for those of you who may not know, the term is described here as the microorganisms in a particular environment. And that could be not just on the body, but also in places like the sea or in aqueducts or in sewer systems. And so they're very important to know the microbiomes, because what it is the microorganisms that can actually exist based on the environment. So not just the food source, but the temperature and the humidity and such. And so if you want to know more about the specifics of the actual skin microbiome, which we'll talk about today, we're not going to talk about a lot of the basics. But if you scan this QR code here on the screen, you can watch a series of videos we produced called Beauty and the Bacteria. And it goes through in pretty good detail the different basic things about the skin microbiome and why it matters to your skin health and your skin beauty. So here, when we look at the composition of the microbiome on the skin, what we can see here by the different colors represented here on the left-hand side is the different colors represent the families of bacteria. Where on the right-hand side, the bar charts, the colors represent actually genera, which a genus species. So it's not yet the species, but it's the genera that is the order above that. And so what we can tell from looking at this is that on the right-hand or the left-hand side here is that from place to place in the body, there's unique compositions of the types of bacteria and other microbes as well that exist on those locations. And interestingly enough, there's more similarity of samples between person to person on the same exact area of the body than there are between the different body sites. And this can be seen by looking at the right-hand side on these columns. You can see the first four bars there are the one body part, and the second one is another body, second four another body part, the third four another body part. And you can see, although there are different ratios, the general makeup of the types of bacteria, the genera of bacteria are all very much the same. But if you look between the different groupings there, they're very different. So that is very important to note, because what this tells us is that we can actually use this knowledge to target specific areas of the skin by modulating those specific microbes or using or leveraging those specific microbes in that area. So here's an example of how we might do that. So what we do know is that over these different areas of the body, we can see these different genera here that are composing the microbiomes of those different areas. And what most of us can see here is that it's very blue slide. And so most of the composition here is actually Propionibacterium, which is the genera that contains Propionibacterium acnes, formerly now called C. acnes or Cutibacterium acnes. And so that means that that's the majority of what is on the skin in these areas. But over time, as we age, it's well known that the two most prevalent genera, which is Staphylococcus and Propionibacterium, or Cutibacterium now, actually reduces significantly. Now, it's pretty much understood that the reason for this reduction is due to ecological changes in the skin. So we're producing less oils, therefore, and our skin's drying out. There's many different changes that occur as we age that actually make the skin less hospitable for some strains or species of bacteria and more hospitable for others. So what we also know is that if you look at this as well, you can see that the diversity actually increases. So the types of species, or the number of types of species of microbe actually increases as we age. And it's one of the things that we want to kind of put into everybody's thoughts as we're speaking through the clinical evidence today, is that there is this kind of mantra in the field of the microbiome, even in a lot of the clinical literature, that a more diverse microbiome equals a healthier microbiome. And a lot of that comes from the gut health studies, where they sample guts and show that more diversity is more equated to healthier states versus dysbiotic states, which have less diversity. However,
Thomas,
while that may go ahead.
Oh, sorry to interrupt you, but I just want to thank you so much for inviting me to join you. I just find this to be such a fascinating topic, and I've thought it was fascinating for so many years. And I think it's so beautiful what you're doing here is you're showing us that we always think in terms of pathology so much, or I know I've always been trained at what's the problem? What's the bacteria that's overabundant, or the yeast or fungus or whatever that's causing the problem that's presenting in my clinic? And we saw on that previous slide with the body that you have all these different worlds on your body of the warm, wet areas and the cool, dry areas. And we know this, right? And as dermatologists, especially, we're always talking to people about how to maintain different sections of your skin. You may talk about body folds in a completely different way than you talk about distal extremities. And it makes sense when you see, wow, there's a completely different population of microbiomes there. And on the current slide, it's not something that I had thought of before, which is with age, We know things are going wrong. We know things are going wrong because, as you said, of ecological, whether it's UV or pollution. But it's really fascinating to think that as time goes by, it's not just your skin, it's the organisms that are living there are also being impacted. So I just think that introduces this whole paradigm shift that we can be thinking about what does it mean to have healthy skin. So I'm super excited to dive deeper into your science.
Well, we're about to dive deeper. And so, one of the things that you just mentioned, which is just age, but also if we look at other disease states, like for instance, dermatitis. So it's pretty much well known that dermatitis has an association with certain bacteria that you just alluded to, the pathogenic ones, right? But one of the things that you also alluded to is that we don't really think about what are the good ones, right? We think about, okay, let's get rid of some. We don't think about what happens if we get rid of all. An example of this is when I was in my postdoctorate, I was put on some pretty hefty antibiotics because I had a skin infection. They didn't know what it was, but before they cultured it, they just gave me this really potent antibiotic that I had an adverse event or adverse reaction to. And so they took me off of it, but they didn't think about the fact that they had just wiped out massive amounts of my good flora. And I ended up hospitalized with not only a staph, a MRSA infection, but then I also had C. diff and pneumonia, all because you basically killed off a lot of what protects us, not just the bad stuff. So, very much when it comes to aging like this, when we think of the parallel in our disease states like atopic dermatitis, not that we want to really equate aging with a disease, but atopic dermatitis, we know that, for instance, there's a lot of Staph aureus that's blamed for the lesions of atopic dermatitis. But also what most people don't realize is that we also know that C. acnes is less present in those same lesions. Now, we can go into some detail as to what that would mean. We're not going to today because we don't have the time. But when we look back at aging here, we ask ourselves the question, okay, so if while we're aging, we get more diversity, we see less C. acnes and Staphylococcus, and more diversity is supposed to be equal to more healthy state, would we really equate aged skin to healthier than young skin? So that's a example where in eczema or atopic dermatitis, there is more diversity in healthier non-lesional skin than in lesional skin. So in some cases, yes, it is true more diversity equals healthier states, but that is not a blanket statement we should make across the board because it's not always true. Additionally, in skin, there is multiple studies that have shown that in the hair follicle, more C. acnes is equated to healthier skin, where more diversity and ingress of other strains is equal to more incidence of acne. And so that's another case where diversity doesn't always equal healthier state. So why does that matter? Well, and what can we do that basically would counter this? And really quick before I move on, I did have to make one more comment about this, which is, so the question is, if ecological changes in skin while we're aging is associated with a change in our microbiota, a loss of certain microbiota, the real question is, if we restore that microbiota, can we restore the condition of the skin? We know they're associated, but is it, something that we can probiotically change by placing those bacteria that are lost back on the skin? A lot of that is being done for disease states like atopic, where they're trying to find some strains to put on to displace the bad ones. But can that be done for all these things, like for aging or general cosmesis? So, right.
I mean, you're saying is aging a disease state? Like, that's
exactly.
Super cool. Like, it's just fascinating. Fascinating. Is aging a disease state that we can manage by managing the microbiome? So that's cool.
Well, I guess when we're being politically correct, we want to say no, it's not, because everybody ages and they want to feel comfortable, in their skin, and we don't want to discount people just because we're aging. We're all aging. But when we think of it from a physiological perspective, what is a disease state? And yes, aging, there's a lot of hallmarks that are similar between aging and things like cancer. So there's a lot of similarities in the way that the body functions or dysfunctions that we can actually mitigate if we treat them similarly and in our interventions. And so probiotics is well known in the gut for an intervention for things like IBS and other diseases. And so when we think of it for skin applications, we really have to ask ourselves, I mean, it seems kind of silly, but we have to ask ourselves, what is the definition of a probiotic? Because you'd think that most people would know, but unfortunately, there's a lot of more or less bastardization of the term because it's used a lot in marketing versus its actual scientific meaning in order to get the buzz, because it's a very hot and trendy thing. I'm not sure, Dr. Hooper, if you see that as well, if people are asking for it or whatnot. But there is a lot of buzz around it. Yeah.
Oh my goodness. This to me was my biggest hurdle. When I first met you and started learning all of this, my first hurdle was thinking of all the people who show up in my office telling me they're taking a probiotic for healthy skin. And I know just from reading and learning through the years that all of these oral probiotics are based on GI, which is not necessarily translatable to derm, probably related. They're based on fecal studies. They're not based on what's good for skin. What the heck are you taking? Which is the one you're supposed to recommend? And so I think dermatologists and physicians have gotten a little bit like leery of this word probiotic because it's such a giant word, as you show here. All a probiotic is basically a bacteria. It's not necessarily, is it good, is it bad, depending on which definition you read. But I think this was probably my biggest hurdle in thinking about what is probiotic skincare, because there's certainly been a lot of buzz and my patients bring me things that they say it's a probiotic. And I say, well, what the heck bacteria is it? And if it was healthy for fecal, does that necessarily mean it's great for your face? So I think you're hitting such a hot button here that it's such a trendy term. It sounds so healthy. People are so into using the power of the microbiome and using probiotics, but we kind of don't totally know what we're doing here. And so that's where what you're doing in these slides tonight, I think the audience is going to be as surprised as I was to learn some very specific things that matter when you're talking about what a probiotic you were using.
Yeah. So when we talk about those gut ones that you're talking about there, it's one of the things that we don't want to discount everything. So there's no like dogma here to say there's no gut probiotic that could be useful on the skin. But what we're going to do today is kind of challenge that concept of, well, there should be certain things that a probiotic should be in order for it to actually impart benefit. And so if we look at the term here, the definition of the term probiotic, you can see that the common vernacular is very centric on the GI tract, because that's where the most emphasis has been placed up till now. However, so if you read that there, it has to do with an example of lactobacillus for the digestive tract. But the thing is, our digestive tract, while it is connected to our skin, it's not our skin. And so what grows in the digestive tract is not going to grow on the skin necessarily. And we'll get back to that in a second. But lactobacillus, for instance, if you look at most of what is called probiotic skincare, most of them have either what they claim is lactobacillus strains in there, or they have lactobacillus ferment, which the ferment can actually be useful. But the strains themselves, if they're even alive, the question is, can they truly engraft onto the skin? Let's put a pin in that real quick before I want to get to these other two definitions, which the World Health Organization gets a little bit closer to a definition that is more acceptable, which they call it the live microorganisms, which when administered in adequate amounts confer a health benefit to the host. So that's actually correct in the sense that in order for it to confer health benefit to the host, you have to ask yourselves the question, what has to happen for that to happen? And we'll get to that in a second after this last definition, which is probably the worst definition, but unfortunately it's the one that is being composed, or at least proposed, by the International Cooperation of Cosmetic Regulation, who are the people that are bringing the products to the market, right? They're the ones that are telling us how we should label certain things. And they're calling a probiotic a live or dormant microorganism, which is a horrible definition, unfortunately, because then that could be anything. It could be flesh-eating bacteria, because that's a live microorganism. It could be Staph aureus, which maybe there are some strains that we'll find out are beneficial. But right now, what if it's a pathogenic one that causes atopic? So what if it's MRSA? These are things that we would never want to put on our fence, nor should we. And in that sense, all these definitions together become very nebulous, so that a marketing brand could basically put out something that they call probiotic, when in essence, there's nothing probiotic about it. There's nothing healthy about it. So what should we consider a legitimate probiotic? We have to lean on the scientific literature where, there have been a lot of great minds that have thought through this. And basically in this publication that's cited here below, this particular set of authors came up with these sets of criteria that would make a legitimate probiotic, whether it's on skin or gut or anywhere. It should display a high resistance to the stressors specific to that location. So if it's on the skin, it has to be able to withstand the stresses of skin, whether that's the UV or whether it's the pH, whether it's the oiliness or dryness or the moisture, wherever it is on the skin. And that's one of the reasons why we have differences from site to site on the skin, because there are different conditions. It's also the reason why lactobacillus, unfortunately, is not going to thrive very well on the skin. Now, interestingly, it does thrive in the vaginal canal, that's what it's, where its niche is. And more interestingly, is that on the skin, it's actually more associated with disease states to have lactobacillus on the skin. Again, old age, you tend to have more lactobacillus on your skin than when you're younger. And so it's not typically something associated with healthy skin. Additionally, it has to have the capacity to survive in that relevant area of the body. So it has to be able to engraft and survive. So if it can't engraft onto the skin, it's not something that's going to be able to impart any amount of health that is going to be quantifiable. And then the rest of them are things that are not necessarily the focus of today, but it has to lack any transferable antibiotic resistance genes, clear benefits through modulation of other microbes, therefore keeps the bad ones at bay and encourages the good ones, and then be non-pathogenic, non-toxic, and provide protection against disease-causing microorganisms. So in order for us to have a legitimate probiotic, all these boxes need to be checked. But it starts with the first two, because if it can't engraft, it can't do the rest.
So I think, do you mind just quickly going through prebiotic, probiotic, postbiotic, because that's sort of confusing, and alive is sort of unsettling, but it's what a probiotic is.
Well, I mean, most people, when I tell them, my mantra for years has been, you have billions of bacteria on your face, and I think that's awesome. And a lot of people kind of cringe at that because we're taught from a very young age, in my generation was the Mr. Yuck stickers and, all that stuff where, we're taught that germs are bad. But in essence, they're always on us. We do not live in a sterile world. And there's a lot of evidence that shows that when we're more dirty in our habits, we actually have less incidence of inflammatory disease. And so our bodies have an immune system that's made to cope and actually be in balance and harmony with our microbes. So the prebiotics is actually, a lot of people think of them as the food for the bacteria, but it's also the environment, the proper environment. So prebiotic should be something that's conducive to allow a bacteria thrive, whether it's a food source or an environment. A pH can be considered prebiotic in a sense. But the problem is that a prebiotic also you have to think about if a prebiotic is supposed to feed a probiotic and it feeds also pathogens, is it truly going to be considered a prebiotic in that sense? Then you could get, there's a lot of kind of nuances because you could say, well, if the probiotic is actually going to be as powerful as it should be, it shouldn't matter if you feed everything because it should overpower the bad ones anyway. So that's where, but in the simplest form, a prebiotic is something that would feed the probiotic so it could do its thing. Now, a postbiotic would be considered the metabolites of the probiotic. So a lot of people think of it as a waste, but it's not, because some probiotics make things like vitamins, and they do other things like they make enzymes and such. And a lot of times it's kind of a symbiotic thing where they don't do it necessarily for our benefit. They do it for their benefit, and it just happens to benefit us as well. So an example is with C. acnes, and we're going to talk about it in a second, is they make antioxidants because they're facultative anaerobes. And so to protect themselves from the oxygen that's around our skin, especially on the surface of the skin, they produce these antioxidants, and that also protects our skin. So it's a symbiotic thing. And so it's not just waste. So don't let anybody tell you it's the poo of the probiotics, because it's not. It's actually, there are some there are some things that are byproducts of metabolism. Propionic acid, which is a great molecule, is one of them. because it digests triglycerides and makes these short-chain fatty acids. But there are other things like enzymes and proteins that are secreted that are not waste products. They are things that are secreted for a purpose.
So I just think we all accept the fact there is a microbiome. We have billions of bacteria all on our body. And you're telling us these bacteria or these probiotics are secreting postbiotics. They're secreting something. And if you think about all the things we're applying to try to change our skin and try to make ourselves get out of a disease state or look better. But if you think that you currently have live organisms secreting something, you probably want to control which live organisms and what they're secreting. And I know we have slides on that, but I think manipulating your own microbiome is just a game changer.
100%. And let's talk a little bit about what those microbes are that we might want to curate. Because, there, like we've talked about, there's a few that are the majority of what's on the skin in any given area. But on the face specifically, in any sebaceous area of the skin, we do know that C. acnes and Staphylococcus species make up a bulk, along with some corneoforms. But when we really think about, okay, what would make the best candidate for a skin probiotic? So we know it has to engraft on the skin. We know it has to be able to survive there. So the best candidate is going to be something that already is there, but just curating the very best strain of that particular thing, or that particular species or that particular genus. And let me tell you, we're going to get into it in a second, but strain really matters. And that's one of the reasons why curation of the microbiome is going to be a huge deal in the coming years, because we don't yet understand. A lot of people think of species as one thing. For instance, when we say C. acnes, most of the people on this call probably think of acne. They think it's the cause of acne, and the fact is, and we're going to show that actually some of the strains, yes, but some of the strains, absolutely the exact opposite, to where someday I pretty much believe there's going to actually be different species made between these subspecies of C. acnes because of the significant differences. But when we look at skin, we do know it's not a flat sheet. And this is something that Rich Gallo published several years back, where he did a calculation that showed if you take the surface area of the skin as it's typically thought of, it's about 2 meters squared. But if you include the calculation of all the surface area of the epithelium of the follicles, there's actually an order of 10 times more surface area of the epithelium in the follicles than there is on the surface epithelium. So that means that the bacteria that is in the hair follicles is actually 10 times more access to the skin and the immune cells that are underneath of the epithelium than anything that's on the surface. So we really need to pay attention to what is both on the surface, but more so what's in the follicle. So when we look at the skin, this is another study by Rich Gallo where he showed that if you look at a cross-section of skin that has a hair follicle versus one that doesn't, interestingly enough, although we thought that the epidermis and the stratum corneum were a barrier for keeping germs out, unfortunately, no. There's bacteria all the way through our tissues, although not in huge amounts, as you can see on the right side here. Look at the scale, please, because you do have some bacteria there. But if you look at the hair follicle, you have orders of magnitude more bacteria in there because it's a conduit for it to go into and engraft within. And as you can see here, as the deeper we go, the more bacteria that we tend to have. And one of the reasons for that is because the major component of the microbiome of the follicle isn't facultative anaerobe. And you're probably thinking exactly what it is Cutibacterium acnes. And that's what the whole talk is about. And it's the reason why we chose Cutibacterium acnes to study as a potential vehicle as a legitimate skin probiotic. Initially, I was searching for something that could actually deliver medicines to the different tissues. So I chose Cutibacterium acnes because I wanted something that would not just hit the surface, but also would hit the whole thickness of skin. And the hair follicle, of course, transverses not only the thickness of the skin, but goes into the subcutaneous fatty tissue as well. And so this is a graph that was made by Dr. Lee at UCLA. And what it shows us is that these are all individual follicles from people on the right-hand side that have no acne and the people on the left-hand side that have acne. And we can see that these individual follicles actually have, when you put them all together, about 89% of the follicle is composed of C. acnes. And so the whole idea that C. acnes causes acne is absolutely false. It's not just this individual species. It's also the genetic predisposition of the person. But more so, when we talk about C. acnes, what is more important even than that is that it's not just a species. It is a number of thousands of strains. And some of these strain groupings are so genetically different that they've been put into separate phylogenetics groups. And that's why we call them of 3 distinct subspecies now, because they're so different, they couldn't just call them C. acnes anymore. So a lot of publications in the past that called out C. acnes for doing this, that, and the other thing did not realize that if you use different subspecies, you'd get very different results. And so here's a series of the 10 most prevalent ribotypes, which is a type of way to designate a characteristic of a given grouping of a bacteria. And so you can see here underneath this red box, which is the type, this is the ribotype that we actually isolated. So not all, RT6 is not a strain, it is a ribotype, meaning there's many different strains in RT6. But you can see that the RT7 and up to 10 have almost complete association with acne, meaning those are probably more pathogenic types of C. acnes, where RT6 and other of these ribotypes have lower association, where RT6 has almost no association with acne. So then the question becomes, oh, go ahead.
This slide is mind blowing to me, just because it is so shifting of what we've all been taught, that what causes acne, and you have your things, your follicular plugging and your hormones and your C. acnes. And I mean, you don't even think, we are lumping C. acnes, and there are like criminal C. acnes out there that are just disasters. And if you have those strains, those ribotypes, you're in trouble. You're going to get acne. But if you have this RT6 that you have in a box, 1% of people with acne have that one, and 99% of normal people have it. Like, that's the one you want. And no one told us this before. So I think this is sort of the most important, taking my mind away from that whole lumping of probiotics into the curation of the real science, the real hard stuff that you're saying, no, we see it right here. This is the ribotype that you want, because you're going to have C. acnes. You showed us that. Everyone has C. acnes, but you want the right guys. So I just think this slide was awesome.
Right. And with any probiotic, whether it's gut or skin or whatever, most legitimate scientists will conclude or tell you the benefits or detriments are going to be very strain specific. So that's why some companies are very private about their strains and isolating their strains and stuff, because the benefits, it takes a long time to characterize a given strain and identify which strains cause benefits and such. So that is very important, is considering is the strain. Now, again, why does this all matter? So the one thing that I want to say or remember is that C. acnes is ubiquitous. Everybody has it. You cannot get rid of C. acnes. You're going to have C. acnes. And so to Dr. Hooper's point, you want the right type of C. acnes. And let us tell you why. It's not just going to take up space. It actually has a very real response. So in this particular study here, these are actually from 2 different papers, but I combined them because the idea is the same, where on the left or on the right-hand side here, basically, they just showed, and I can't take too much time because I am kind of running over here, but they took basically pH is indicative of a healthy C. acnes, which is associated with health, meaning they obtained it from a healthy individual, where PA is a C. acnes they have obtained from somebody who has acne. And on the top graph there, they actually did this. What they did is they took the healthy C. acnes and they put it into a broth where they grew it, and then they took that ferment product and grew immune cells in that product or in that ferment. And it caused those immune cells, when they put it into a petri dish with acne, C. acnes, they obtained from acne, it actually killed them. So that downward trend there is because they saw a reduction in the acne-causing C. acnes. When they did the opposite thing, where they took the acne-causing C. acnes ferment and they put the immune cells in there, it didn't kill the acne C. acnes. So what this shows is that the healthy type of C. acnes actually has an immunomodulatory function. So on the right or left-hand side here, you can see this graph where actually the C. acnes comes in contact with things like macrophages, sebocytes, and keratinocytes, and actually can induce certain growth factors. The key one, which is IL-12, which can actually cause a CD4 naive cell to go of 1 of 2 lineages. It can either become an Th17 lineage, which can actually cause homeostasis or quiescent skin, or it can become inflammatory Th1, Th17 phenotype. And that is what is associated with acne. And so you can actually modulate your CD4 positive cells depending on the strain of C. acnes that you have. So that's another reason, number one reason why we should never try to lump them in as the same thing, because they absolutely have different modulatory functions. Now, one of the things that they have in common is that they produce propionic acid. We already talked about this. And here's a graph of a strain that we actually produce called XYCM42. And you can see through the growth curve of the bacteria, this is actually in production in a 200-liter fermenter, you can see a huge amount of propionic acid is being produced. That's why they used to be called propionic bacterium acnes, because they produce a large amount of that short-chain fatty acid. So why is propionic acid important? Because these C. acnes are producing it all the time on your skin. And it has been shown that you can actually see that propionic acid here can reduce UV-induced melanogenesis. So this means that there's actually a function through tyrosinase inhibition, the same as a vitamin C would do. But remember, these are on your face producing it all the time, where vitamin C, as we all know, is a molecule that is very labile. It can fall apart with heat, oxygen, light. That's why we use them in the dark bottles. But we all know it works really well. And so not only is it an antioxidant, but vitamin C I'm talking about, it's an antioxidant, but it also reduces pigmentation to even out our skin tone. But now we're finding out that the things we were trying to kill actually do the exact same thing at the exact same levels as a vitamin C. Here is an example. The second here is it reduces biofilm. So ferment of a C. acnes actually can reduce biofilms produced by Staphylococcus species. It can actually reduce or even kill Staph aureus species based on which strain you're talking about with C. acnes. And this is the one that I get the most excited about, is that, is that not only did, what happened there? Did I skip one? So let's see if I can keep it. Or if I don't, I'm just going to let it go. But not only did okay, I'm just going to talk about it. Not only did a group back a few years back, I think 2017, found a molecule called ROXP, which is unique to C. acnes. It's an antioxidant that is potent, just as potent as vitamin E and vitamin C. And we were actually able to show that if you apply a certain amount of these C. acnes that we isolated, you can actually produce upwards of about 15 milligrams of vitamin C equivalent antioxidant activity on a daily basis. And so, of course, that is not all at once. It happens throughout the day. So it's manufacturing it constantly throughout the day. So, but that's very interesting. But also what we found was that if you remove the ROXP, that isn't the only molecule. So there are other molecules that we're still characterizing that are antioxidant as well. So there's just as much of other antioxidants than there is ROXP. So there's a huge amount of antioxidants that these C. acnes produce. Now, this slide here that apparently fate wanted me to talk about because it wouldn't go away is actually something we did in our own lab. And this little dotted red line is showing you where the control levels of these inflammatory gene signals are typically. The big bar in the middle where it says PGN is where we induced inflammation in these PBMCs, which is basically immune cells, CD4 cells. They induced inflammation with PGN, and then we added IL-10 as an anti-inflammatory molecule, and you can see we've reduced inflammation. And the one on the far right was actually a construct we were making for our therapeutic version, which it delivered an IL-10. It was a C. acnes that delivered an IL-10. We knew that would reduce, and as you can see, it reduced inflammation past baseline. But even more interestingly is the XYCW1, which was our wild type strain that we currently do research on. It actually reduced the baseline as well in normative PBMCs. So what this shows is that not only is it not an inflammatory bacteria, which other strains have been shown to be inflammatory of C. acnes, it actually is anti-inflammatory. It inhibits inflammation. And that's why we looked at this checklist of what the scientific community felt a true legitimate probiotic should be. And the C. acnes subspecies defendens strain XYCW1 or XYCM42, which is the strain that we use in our clinical research we're about to show you, actually ticked all the boxes. It engrafted and it produced benefit on a consistent basis. And so that's why we chose it. And because we're a little bit over time, I was going to stop and take some questions, but let's just move on to the clinical part, because that's the part that I think is the most exciting. And I do want to allow for some time at the end for some questions. So Dr. Hooper is going to kind of go through the clinical portion here, and I'll add some commentary as needed.
Okay, great. Thanks for turning it over to me. And I think this is, where it all hits the road is that we always see, and with skincare companies, they'll show us these cool basic science slides and things that happen in a lab. So what we've seen is it looks like that you've identified a strain of C. acnes that is going to produce antioxidants, decrease inflammation. It looks like it's just going to be a little workhorse for our bodies. And so what I'm going to review now is the data of the clinical study where they looked at the efficacy of applying this strain and seeing what happened to people. It was 121 studies enrolled for 8 weeks. These were normal skin. We were not trying to treat any disease state at this point, but just normal skin, just to see what happened when you applied the product. And so on the next slide, I don't know if I'm supposed to advance them, but I can. Okay. So on the next slide just goes through the study design. So they got a little kit, right? They got a prebiotic cleanser that was just going to set everything up. As Thomas already told us, that prebiotics are just setting up an environment that will make it most healthy for the bacteria that we want to have on our skin. In the morning, they would apply this ferment-based serum. So you saw that the ferment, the path on the reports on the ferment, and that the ferment itself was the one that reduced melanogenesis, that also improved inflammation, improved short-chain fatty acids. So it looks like a serum, the product of the bacteria, and then moisturizer and sunscreen. And at night, just a gentle cleanser, and then the live gel. So the live gel has the live strain in it, and it's freeze dried. So you missed on this prebiotic activator to sort of wake up the bacteria. So it's a probiotic, customized regimen for your skin. And no, they didn't use anything else. And they did questionnaires. So questionnaires at weeks 1, 4, and 8, and daily diaries. Broad range of age groups, mostly white, but all skin types included in the study. So once again, this was just sort of seeing, what does this product do when you just put it on humans? So important. We always think when we're trying out skincare products, the first thing is we certainly don't want them to irritate our skin. And a lot of anti-aging and anti-acne products can cause irritation, and just all kinds of skincare products can cause irritation. So this was just a nice Zycrobe regimen is what this is called, and it didn't negatively impact any of the normal skin properties. So the transepidermal water loss was great. No loss of hydration, no irritation over the whole time. Hydration actually increased a bit over time. The skin was better hydrated because the product was tolerated very well. And maybe that prebiotic sort of set things up for that. And this is nice. Erythema went down. So sort of going over and over that this is not going to irritate your skin. This is going to be something that's going to hopefully decrease inflammation. So we start translating what do we think inflammation that we see in the earlier slides that are the basic science of interleukins and all the interferons, but they translate to inflammation, and erythema is one of those signs. So erythema went down over the study. And then this is something, people come into my clinic all the time and they say, I just don't want oil on my face. I don't want to be oily. I don't like my pores. And I think every dermatologist I know just dreads hearing that conversation. And, you tell them, you try to sort of cheerlead with them and say, oh, but you won't have so many wrinkles because your skin is more moisturized, but they don't like grease and oil. So this was a nice drop in sebum production, not just on the nose, but also on the forehead. Okay, so now this is just gets into...
Dr. Hooper, I should comment on that slide really quick because I didn't include that in the science background part of it, but there is actually several publications that talk about C. acnes actually modulating production of oils, but it also has been shown to increase IGF-1, which can increase ceramide production and help with the barrier repair. And so that's one of the reasons why we think it's associated with or not associated with dermatitis, because we see a lack of C. acnes in dermatitis lesions, which we know has issues with barrier. And so I just wanted to mention that because when we think about reducing sebum, There's probably multiple functions of that, which is not only is it modulating the sebum, evening out the sebum production, but also it eats it and produces short-chain fatty acids. So some of the reduction is because you have all these microbes now that you put on your face that's eating all that oil.
So that's, yeah, you showed us how deep they go, all the way down into your 25 square meters of skin with your appendages. And so those oily pores that everyone's stripping and everyone's squeezing, we're just going to apply this and it's going to dive down there and actually eat up the sebum that we don't want and produce some antioxidants and anti-inflammatory factors that we do want.
Right, and we didn't show it in this deck, but if you go back to our webinar from last month, there are some photos of it where the first study that we did about 5 years ago, I was the first person to put it on my face because I didn't want anybody to do it. I wanted to make sure it didn't cause acne. I was a little bit scared at first. But when we did it, we actually had a guy that came in that had a lot of blackheads, and we weren't anticipating anything because we just thought it would help to balance the microbiome. But it actually dissolved the filaments of the pores, and you can see it on the photography, and you can see it on the image if you go back to that webinar that's on the website. But also because of all the antioxidants, it reduced the amount of oxidation of any filaments that were left. So you didn't see the blackness because it wasn't able to oxidize. And so that was an unexpected result, but it's interesting that I continue to observe that in individuals, is that it tends, because the strain eats that sebum, it reduces the filament, and then there's a lot less of the oxidation.
And it kind of, it makes sense when you go back to the basic science that you're going to be impacting these small molecules and small chemokines and interleukins, and seeing what that does clinically, I think is fascinating. And the other thing that's nice is how quickly people saw some improvement. This is the percentage of people saying they were improved. And it was lots of people, 80+% at week 1 saying, you know what, things are looking better. My skin is healthier. Looking at wrinkling and overall photo damage, these were questionnaires administered. Seeing improvement.
Actually, these are actually the clinician grading. So the questionnaires aren't included in this study, aren't included in this deck. These are actually the clinical grading. So this was actually when yeah, these are from the photographs. Yeah.
These are from the photographs. Sorry about that. Yes, these are from the photographs. So an expert investigator looking and saying, do I see any improvement? And significant amounts of improvement seen. I don't talk to my patients about looking really at skin quality at 4 weeks with anything that I do for them. Everything usually takes several months. So this is really, to me, heartening to think that you may start seeing some improvements from an expert's eye in photography early on. And once again, this kind of gets more into the fun, the pretty words. Is the skin more clear and even? How's the mottled pigment, and how's the discrete pigment? And these were things that you can see. Each column of graphs goes through each quality, and so you're seeing improvement graded by the physician in all of these things. Improvement in dullness, improvement in unevenness. Pigment, not quite as exciting, but again, very early, very quick studies, just in the first 8 weeks. But you're seeing improvement. And to see numbers like 20% of patients have an improvement in pigment at week 8, I think that's very impressive, because as we know, things take a while in dermatology. This was fun. These are another term that gets my day a little bit longer, is when someone says, what do I do about my dark circles and my puffiness? Because they're multifactorial, as we know, and there are always combination treatments needed, but everyone wants a cream to help with their under eyes. And this was surprising to me. And we can see in some of the pictures in just a couple of slides, this wasn't even something that I'm glad you put it in as a questionnaire, because I hadn't even considered it. But the clinicians were grading good improvement. Lots of people had improvement in pigment, under eye dark circles, and also in puffiness. And I don't know, Thomas, if you do you think that's inflammatory maybe is the reason we saw that?
Yeah, that's kind of where, I'm with you as far as you have no idea how surprised I was to see some of these things, because even when we look at some of the pictures we're about to look at that are represent these data, I had to ask my Mona Alcom, Dr. Alcom, who's moderating. She's actually the person that was the head of this study, and I kind of had to ask, was this photoshopped? Because some of the results were like not typical. And so then I started seeing it, patient after patient. It started to ring true that actually we were seeing something very real. And then when we got to the under eye dark circles and under eye puffiness, that's when I was like, this is, it's more than just, when we think of that, there's more involved in the skin. There's fat involved, there's connective tissue involved. And so I do think that what's happening here is that if you truly have an immunomodulatory function, we can, of course, not equate that to any given product, but from the science that we're talking about, if you have an immunomodulatory function, that's actually going to move the needle on a lot of things, because the way that the immune system functions can actually affect the quality of any given tissue, not just redness, right? Inflammation doesn't just cause redness. It can cause edema, and it can cause, infiltration of other immune cells, which can lead to tenderness. So there's a lot of things that can happen. So to answer your question is, we cannot definitively say what is causing what, but we can say that a topical application of these probiotics and postbiotics from the specific strain actually did cause a pretty big improvement from time point to time. Yes.
Yeah, and we know that inflammation is just so significant and so pathologic in so many conditions. And so even getting into laxity and turgor. And so same idea. Why would you get tighter? But maybe there's some sort of inflammation that's going away. So I want to move on to the slides, because these are what it all matters, about. And these are beautiful pictures of the study results that I really have enjoyed. And the more I look at them, the more I see. But I have no doubt here as to which is the before and which is the after. And why is it? What is the thing? Am I looking at redness? Am I looking at pores? Am I looking at hydration? I think I'm sort of looking at everything. I'd never see pores look much different. I think pores always look the same, but maybe the texture looks better, or pores look the same, but the color looks better. But in this picture, I really see a difference. And I think it comes down maybe to light reflection. You and I were talking about that. Like, could the sebum quality, we always, I know these photographs were so well done and controlled for light and for flash. And so something is making this patient look less red in the cheek, less shiny, less blackheads. And I think some of it might just be the oil that's on the skin, and maybe some anti-inflammatory decrease in erythema.
Right. We'll see later in later pictures that, this is like probably the least impressive of the pictures. And so we'll see later that the redness is real.
Yeah, this is real. I called you up, I know, and when you sent me these slides, and I said, I just need you to show me the lighting and what's going on in the photography studio, because this is flash and it's all standardized. And so, when you meet this woman on the top, I talk to my patients a lot about sort of, I call it just an inflammatory face, whether it's a little bit of sebderm, a little bit of rosacea, a little bit of eczema, acne. I have these inflammatory people. And I tell, even when I'm training my residents, I'll teach them that. I'm like, you can't always put someone in one pot. But if this top woman walks in my office, I'm thinking about, what do I do about her redness? I'm thinking about, what do I do about her oiliness? Maybe her pore size. Maybe she needs some sort of anti-inflammatory on her face. But I definitely get a gut feeling of inflammation. And when I look on the bottom picture, it's just tremendously calmer. I don't know what else to say except the word calm. And I'm going to just keep going. This picture of pore size was fabulous. The pore size, you can't have changed the pore size this much in week 8, can you? I'm not so sure, but with matched photography, and there's definitely a shininess that's not there. And I think that I was trying to decide if I see the wrinkles and the pores more because the shininess is creating a flash. The fine lines look better on the bottom when I look into the infraorbital area. So these are beautiful.
I agree with you. I think, I zoomed in on this one in particular because I wanted to actually look and see if I could match up the different pores to see if they were legitimately bigger or smaller. And I think the oil production definitely, of course, the mattifying effect does reduce the appearance of the pores. It's very hard to actually reduce a pore. Pores, now over time they can, if you reduce the bioburden and you remodel the skin. But over 8 weeks, it's very difficult to actually reduce a pore. But they look slightly smaller, but I think it's mostly the amount of oil there that's reflective. And also, when you reduce the inflammation around the pore, it also reduces the appearance of the pore. And I think that's also what we're seeing as well.
Right, that's a great one. This is not a laser. This is not retinoids. This is not a chemical peel, but would be a happy patient in my practice. Just healthier, pigment is better, appearance of fine lines and erythema looks better to me. I think this was a great one. I think this is a side of her face. So just sort of showing just that same a mattifying effect. And what my patients are talking about all the time now is, I want to wake up like this. I've said that for years, honestly, but I want to wake up like this. I don't want to put a lot of makeup on and mattify and cover. And, seeing this improvement, I think this one, you look at it, you sort of think about the freckles, but when I look at her infraorbital hollow, and she looks young, but the infraorbital hollow looks better to me. And I think that maybe is that edema.
Possibly. I mean, I've said, like you said, on the pigmentation, because if you look at the side of her cheek there near her preauricular area, you can see there's a patch there, and so there's. If you really look, it's hard because she has a lot of freckles. But if you look for a while, you can see that there's actually a legitimate reduction in the freckles, which, especially around the side of the nose there, you can see that some of them are nearly gone. Not that. Freckles are sometimes just genetic, but also they can have UV-induced melanogenesis. And so we wonder whether it's the propionic acid that's reducing some of that pigmentation there.
Right. Acting as sort of an antioxidant and reducing it, like you showed in that basic science slide. Yeah, her pigment just looks healthier. It looks healthier posteriorly in her face as well. This is another one that pigment just looking better. And these are short studies. This is only 8 weeks. So it'd be interesting to see what happens with much longer use, because I don't see most of my skincare patients back so quickly. But I think she has vitiligo, right?
Mm-hmm. She does.
Yeah. So her vitiligo is stable, I think, but all of this pigment that you can see in her forehead and just this sort of muddiness and blotchiness that looks better. And I think her under eye skin looks better too. After I read that slide, I went back and looked at everyone's under eyes, and I thought, wow, it actually does look better. This is her from the side. You can see that giant lentigo, those are a nightmare to treat in every way, or macular SK, whichever it is, and those are hard to treat. And it's looking better. It's looking lighter. I want to get to some of these, and you can even see these fine lines kind of near her oral commissure, these vertical lines that are so hard. Those are also hard to treat. Usually I have to pull out a fractional laser and really try to erase those, and those are looking better.
You can just move on to the same one.
Same one. A lot of erythema looking better in this patient. I think that she looks healthier, not so angry in her skin. Her from the side. I would much rather wake up and have to put makeup on the after than the before, or not put makeup on. And I think, a lot of these ladies are these kind of ruddy, rosier ladies, and I think they just look healthier and better. I want to get to the one.
Well, I don't want to skip. Go to the one before, because I don't want to skip that progression slide, because a lot of people wonder whether or not it's just luck of the draw, but that you just got them on a good day or a bad day. And so this one, I like these progression ones because it shows from time point to time point a progressive improvement in her appearance.
Right. She's not just having a good day. It's not just a flare. And this woman has terrible actinic bronzing and looks just healthier, I think, after. When you look and start picking apart every little fine line, this is not a toxin or a resurfacer or a peel. This is just a few creams to put on morning and night.
Probiotic. Yeah.
Right.
Yeah, and this is the one where if you really start to look at, especially on the below the lip there, you can see that there's areas where lines are completely disappearing, this is one of the ones where I was like, I'm pretty sure you photoshopped this.
Right.
Not just there, but if you look laterally, there's a big line that comes from the oral commissure, and a branch of the wrinkle just is
right
gone.
Mm-hmm.
Yeah. And then, of course, the pores, because of the shininess, is gone. So the pores are very hard to identify in the upper lip. Yeah. So this is this was the first image that they showed me. And I said, this is pretty good results. You don't typically see wrinkles disappearing after 8 weeks, unless you are doing something pretty hefty.
Yeah. It's a beautiful picture. Beautiful picture. Similar results on the other side. This was a nice lady. She had bad acne. Go ahead.
Yeah, this is the one where when you see her from the side, especially the progressive one, this is the one where I didn't really believe the data on the laxity, because I was like, there's no real way you're going to solve laxity with a topical. But when you look at her jowling, not only do you see the acne scar texture improve, but the jowling, I'm like, I don't think it's deniable. It's just such an improvement there. It's pretty amazing.
That jawline is beautiful.
I mean, I kind of like, I rub more on every day just because I want the jawline that she has.
Absolutely. To hold down the body. So I don't want to keep us too late. I want to respect everyone's time. So I'm going to come to this conclusion slide that it all comes down to the strain. And I just think that's a game changer, and that the theory is great, but then those clinical results mirror the science. So I just remember when I was first meeting you a couple of years ago, where it was, why do we need this technology? But it's harnessing the power of of the microbiome to exert effects that we're trying to do with other competing products. So it's just like a whole new entry. And I think you're legitimizing this category that to me has been a little gray and hard to negotiate. So it's all about the strain playing a critical role in symbiosis.
Mm-hmm. Yes, 100%. And, we would love to talk about it for another hour, but we do want to make sure everybody can get to their families. So we have enough time maybe for one or two questions. So Mona, if you want to pop on and ask one or two questions from the audience.
Yes, sure. So Dr. Hitchcock, a question for you. Is Dr. Hitchcock aligned with the reclassification of C. acnes to P. acnes?
From C. acnes to P. acnes? Is it P as in Paul? Did I hear that correctly? Okay, well, I have not heard of a reclassification. So if that's happening, I need to go look at that. So it used to be Corynebacterium acnes, then it was P. acnes. Now I believe it's Cutibacterium acnes. So if it's been reclassified, I am not aware of that. So I'll have to look into that. I'd have to know the reason for the reclassification, and also whether it's the whole species or one of the subspecies.
Okay, another question. What other strains of bacteria would be considered legitimate skin probiotics?
All right. I mean, I don't know if Dr. Hooper, if you have any ideas on that, but what I would say to that is I'm sure there probably are other strains and even other species, but we should not venture to guess because, again, really you have to characterize the strain that you are going to be proposing in order for you to answer whether or not it's a legitimate skin probiotic. If we were to speak blatantly, it would contradict what this whole talk is about. And because if we were to say a whole species or a whole subspecies was to be appropriate, it would basically negate what we just talked about, which is that the benefits or detriments of any given species is going to be, or any given strain is going to be strain specific, and you can't just equate it to any strain within that species.
Okay, great. Thank you, Dr. Hitchcock. A question for Dr. Hooper. Dr. Hooper, do you need to use both the probiotic and the postbiotic, or can you just use one or both?
I assume that is referring to the system, where you have the several steps in the morning and the several steps at night. All we have so far is the clinical data using the system. So the system includes prebiotic and the spray to activate. So at this point, I would recommend using the whole system when it is available. I think as dermatologists, we're always using things in all sorts of ways. But I think right now I would stick with the science, go through the steps. And I think it is just fascinating. And I think to that earlier question, we don't, as far as I know, no legitimate strains, but I bet they're out there. And there are those people that you've always seen that why is their skin so gorgeous that I don't think it's just Pond's cold cream every day. They probably got some genetic benefit from their strain of bacteria. So this is just the beginning, I'm sure.
I agree.
Thank you. Thank you both so much. So this concludes this evening's webinar. Thank you to both of our speakers, Dr. Hitchcock and Dr. Hooper. And for those of you joining us this evening, should you wish to further your education on skin microbiome, please follow our Beauty and the Bacteria docuseries on the Crown Laboratories website. On behalf of Crown Laboratories, have a wonderful evening.